All right, so for the past month and a half, 2 months, something along those lines, I have been working on a very big project with everything going on in the world with the SARS-CoV-2 pandemic. So basically what I've been doing is pulling a wide spectrum of people who are in high school to people who are senior citizens. I've also been polling people with various degrees of familiarity with science-related topics, people who have absolutely no science background to people who are PhD students. I compiled all of these questions into a nice, concise list of topics, and I reached out to my friends who are in fields relating to viruses, the spread of infectious diseases, and the immune system. And I also reached out to some professors at Cornell in order to help put together this nice 3-part video series. Now, what is this video series? The way that I wanted to structure this video series is to basically give the first portion of all of these videos to a graduate student, where the graduate student will explain a concept that is related to the pandemic that's going around us. Once the grad students are done explaining their portion, I then go on to a Q&A session with 1 or 2 Cornell faculty in a field related to that topic, and I ask them some of the questions that people had provided me. So with all of that being said, let's go ahead and jump into it. This first video is focused on virus biology. 2 big disclaimers that I want to make before moving on to the actual content of this video. The first one being that all of these recordings were made and provided to me about the end of March. But then the second thing is that nobody in this video works with SARS-CoV-2. It's not their main focus of study. Everybody who is in this video works with a topic that is relevant to the current pandemic going on. So to start us off, my friend Robert Lopez, who is a graduate student, will be presenting a little bit about kind of the structure of viruses and a little bit more about the coronavirus. So Robert, take it away.
Hi everyone, my name is Robert Lopez. I'm a 3rd year PhD student at Cornell University, and I'm currently working with Dr. Colin Parrish, specifically working with canine parvovirus. And I'm here today to talk a little bit about viruses to better understand the pandemic that is going on worldwide with COVID-19. So I hope you enjoy it. So if you go to the internet and look for virus, there's so many different definitions out there because viruses are like that. They're very complicated in the ways that they eject a lot of different functions. They're so diverse, and also they have different mechanisms to hijack the cell machinery in order to replicate. So viruses, I'm going to define it as a genetic identity that has the ability to transfer genetic material, either DNA or RNA, from one cell to another. All viruses that exist, they need to infect and get inside the cells in order to replicate and form more viruses or progeny. Viruses are everywhere in our air, and they can infect any form of life. So they can infect mammals, such as humans, dogs and cats, but they can also infect bacteria and archaea, and they can live in different conditions, and they can adapt, evolve rapidly, and also jump from one host to another. But not all viruses cause disease. Some viruses have the ability to turn off genes that are associated with other disease and stop that disease in humans. They're good for our bodies. So let's talk about the structural aspects of viruses, because oftentimes the way that these viruses are arranged and the structural aspects that they have, it can be useful for better understand the way that they behave, the functions that they eject, but it could be also predictable of any pathology that is associated with any viral infection. So viruses are pretty simple compared to other organisms, and oftentimes a structure that they have is predictable of the way that they behave. The most simple virus contain 2 different features that are required for any virus in order to replicate and propagate between cells. So the first one is the viral capsid, which is basically a protective layer made out of proteins or a single protein that basically protects genetic material, which is precisely the second component of any virus. So this genetic material could be either DNA or RNA, depending on the virus. And these RNA or DNA contains information that is important for the virus to make more viruses and also to replicate in the cell. More sophisticated viruses have extra layers, for example, an envelope, which is a layer that is mainly composed of lipids, but it can also be associated with other proteins. Other viruses have incorporated proteins that are important for interaction with receptors that are expressed on the surface of the host cell, and once they interact, these viruses can be internalized, get inside the cell, and replicate. So now that we know how viruses work, how they replicate, and some of the structural aspects, we can move on and talk about COVID-19. So COVID-19 is the disease that is caused by the virus SARS-CoV-2, which is an emerging virus, which means that it's a new virus that is infecting humans, and it went to pandemics. So this virus is pretty small and is about 500 to 200 nanometers, which is really, really small. As I mentioned, viruses have two components that are the minimum components for them to replicate. So COVID-19 have these two features, which are the capsid protein and also the genetic material or the nucleic acid. So for COVID-19, the genetic material is RNA, and this genetic material encodes for both proteins that are important for the structure of the virus and also proteins that are important for replicating the RNA inside the host cell. So some of the proteins that are encoded by COVID-19 RNA are the S, E, M, and N. I'm going to explain a little bit about the function of these proteins and their names. So let's start with S. That stands for spike. So these proteins are expressed on the surface of the envelope of the virus, and this protein is important because this protein interact with the receptor that is expressed on the surface of the host cell. So when the spike protein interacts with the receptor, which is ACE2, the viruses can be internalized and release their genome inside the cell and start the replication. The E protein is the envelope protein, and as I mentioned before, an envelope is an extra protective layer that protects both the capsid and the nucleic acid. The M protein together with the S and E protein, they form the viral envelope. And the N protein, which that stands for nucleocapsid protein, is the protein that holds the genetic material inside the capsid, which is the RNA. So the spike protein have been described and it was imaged, and this is important because when we analyze the structure of these proteins, we can get useful information not only for better understand the way that these viruses behave, but also gives us information to better understand how we can develop medications and drugs to prevent the interaction of these viruses with the receptor and hopefully create new drugs that function as antivirals.
All right, so thank you once again, Robert, for taking the time to explain some of those concepts. Genuinely appreciate it. For the second part of this video, I'm going to be turning to Dr. John Parker, who is a virologist here at Cornell. Such a cool guy to talk to. I really had a great time doing this Q&A session with him. He was very enthusiastic about helping me put this video together, getting me in contact with various professors. So I really hope you all enjoy the second part just as much as I did.
So first and foremost, thank you for your time. I'm really excited to hear some of your thoughts on some of these questions that we've gotten from people. If you wouldn't mind, could you please introduce yourself and state your name, your department, and your university affiliation for people?
Sure. My name's John Parker. I'm an associate professor at Cornell University, and I work in the Department of Microbiology and Immunology, part of the vet school.
So, as I had mentioned to you before, we have a variety of questions kind of more in line with the actual biology of the virus itself that I was hoping that you could provide us some insight on. So to start us off, somebody asked, how long do we think COVID-19 survives on surfaces? How may factors such as temperature or the type of surface affect how long it would remain on the surface?
Yeah, that's a great question. So there is one study that's been published already addressing that, and they looked at a variety of different surfaces, including things such as cardboard, stainless steel and glass. And the first thing to say about that study, to bear in mind, is that these are done under experimental conditions. So you can extrapolate a little bit from their findings, but you can't really take that number that they found as being the final word, because as the questioner mentioned, there are multiple variables that you will not know. So, I'll give you an example. Imagine someone ordered a package from Amazon and it arrives in a cardboard box, and they're concerned about SARS-CoV-2 being on the surface of that box. The things they don't know is, well, how long has the package been shipped for? Was the person who delivered it, did they have SARS-CoV-2? Was it ever exposed? What temperature was the package stored at during the delivery process? Was it in the sun? Ultraviolet light will definitely affect the survival of these viruses. So I think rather than giving a specific number, there's some really clear, easy things that people can do. The first thing is that if you're opening mail and you're taking out packages, as you open the cardboard box, when you've taken everything out, wash your hands. And take the packages out, put them into wherever you're going to dispose of them, wash your hands, and then you can wipe surfaces down. Soap and water will kill this virus very easily. So if you follow the guidelines for washing your hands, you're going to be perfectly safe. You can't breathe the virus in from cardboard or from surfaces. It's going to be on your hands. So I think that's the best advice. And I think, if you give people times and dates, then I've seen online people are storing their mail for 4 days, for example. There's no need to do that. You can just open your mail and go about what you normally do, but wash your hands. I mean, you can think about how you might do it, about where you want to open your mail. If you don't want to open it in the house, you could perhaps open it on your deck and then go inside afterwards and wash your hands. But those are sort of simple things you can do.
All right, cool. The next question is, when are people contagious? Is it only when people are showing symptoms, or can people be contagious without showing symptoms?
Yeah, another great question. And I guess we know a little bit, but I don't think we know the complete answer yet. We believe that people may be secreting virus before they begin to show symptoms. There's evidence suggesting that there are people who become infected that don't show symptoms, they're asymptomatic. The things that we don't know are how much virus do these people secrete, if any, and how contagious are they to other people. And based on what we know from other viruses, it's likely that when people are sick, particularly when they first get sick, that's when they're secreting the most virus. Whether they're secreting before they have symptoms, it's harder to know at the moment. But the suspicion is that perhaps they are, and we just don't have the answers to that question just as yet.
So kind of following along those lines of the secretion of viruses, right? One of the other questions that people had was, how do medical facilities test for COVID-19? And is the test sensitive enough to detect the virus at low levels?
That's a great question. So the way they test for this virus is they look for its genetic material. And so we use an assay called PCR, which is polymerase chain reaction, that allows you to amplify very small amounts of genetic material and then detect it, usually with fluorescent dye that can be read by a fluorescence reader. The sensitivity of these types of tests is very high, so they are able to detect very small amounts of material. And so the test is very sensitive, and it's quite specific, and that's the first test they would use. They're looking for the genetic material that's present in the virus. Now, one thing to remember is that if they detect genetic material, the test is positive, but it doesn't necessarily mean that the virus is infectious. So genetic material can be detected, but it may in fact be the person's recovering and is not secreting any infectious virus anymore. So these tests are very useful for diagnosis, but they're not useful for deciding when a person is no longer infectious.
So kind of building on that, just out of curiosity, what factors determine whether or not a virus is infectious or not?
Yeah, so that's a question of virologists. Actually, we struggle to answer that. So I can give you the basics. So this is a virus that has fatty membrane around it, and in that membrane are inserted the proteins that the virus uses to attach to cells. They're called the spike glycoprotein, and that fatty membrane has to be intact for the virus to be infectious. Inside the membrane, the genome is encoded with another protein that's called the nucleoprotein, and it's called nucleoprotein because it's surrounding the nucleic acid. The nucleic acid is what makes up our genes. This particular virus has a strand of RNA, and it's encoded in a nucleoprotein. So even if the membrane's not intact, then the virus wouldn't be infectious. Even if it were intact, not every single genome, that's the term we use for the genetic material inside a virus, not every single genome will be complete. It may have mistakes, and it may not be possible for that virus to actually initiate an infection. So as virologists, we know that a certain percentage of genomes within any population of viruses will be infectious. But it's very hard to predict which ones are and which ones aren't. I haven't seen anything published about that yet for SARS-2, but I'm sure people are going to do that and find out what percentage of the virions that are produced are infectious.
So for the next question, people are saying a lot in the media that this is a virus that probably came from another animal of origin. So what somebody wants to know is, why do certain strains of viruses seem to exist primarily in certain animal species? And then how can these viruses be transmitted to humans?
Let's start from the bigger picture. First thing to realize is that every living thing has viruses that infect it. And that stretches all the way down from bacteria, which have viruses called phages that infect them, all the way up to us. And we know many viruses that infect people. And the same is true for animals. And viruses have been going from one species to another since we evolved. So it's something that just happens. But it's not common that a virus will jump from one host species into another, because once it starts infecting one host species, it becomes adapted to that species. So it's uncommon for a virus that, for example, infects bats to infect humans. But that doesn't mean it can't happen. And sorry, maybe you can repeat the second part of the question.
How can these viruses be transmitted to humans?
So the way that usually happens is we have to have close contact with animals that have this virus in the first place. That's the first step. And then we have to be infected with that virus. And obviously the close contact part comes because humans do get close to animals. If we take the example of MERS, which is Middle Eastern respiratory syndrome. This is a virus that is in the same family of viruses as SARS-CoV-2. This virus actually infected camels, and the first people that became infected were the camel herders who were looking after the camels. We don't really know which species of animal SARS-CoV-2 came from. There's some talk about pangolins being possible, but it doesn't look from the sequences that pangolin were actually the direct transmitting species. But the thought might be that some contact between a human and some other species carrying an intermediate virus is how we picked it up. That's the first step. It's actually really quite common for that initial infection to go nowhere. So one person gets infected, and then they never transmit it on to another human being. And normally when that happens, you might hear about there'll be an individual gets infected with an unusual virus, and then that disappears. So the initial outbreak of SARS, for example, infected a fairly small group of people, so it hadn't really adapted to transmit well between humans. So the virus has to actually not only infect a person, it then has to be transmitted from one person to another. How that happened and why it happened are questions that people are trying to figure out right now. But we know it does happen. We've had examples in the past of viruses getting into other species. So Colin Parrish, who's a professor here at Cornell, this is his area of speciality and how viruses jump from one species to another. There's multiple barriers to that happening, but it happens, and it's just part of the natural evolution of viruses. This is what they do. They infect one species, and then they'll at some point be able to infect another.
And then so kind of following along those lines of ability of viruses to be transmitted from one species to another, some people have cited that there was a report that a dog tested positive for COVID-19. Do we have to be concerned about getting it from dogs?
So that's an interesting question. So the report is true. A dog did get infected. We don't know enough yet. But what it appears to be true is that that infection occurred from a human to a dog and not from a dog to a human. And that is most likely to be the direction it goes in. So dogs may be able to become infected if, for example, they're in close contact with an individual or person that's got the disease. But the other direction, which I think the question is really asking, can we catch it from dogs? There's no evidence that we can at the moment. There's none whatsoever. And it would seem, I don't know enough to really be certain about this, but it would seem unlikely based on the question we had before, how often do these viruses jump from one species to another? Now we're talking about going from whatever animal that this virus was infecting into us, where it's now spread. And now we're talking about going from humans to dogs and spreading amongst the dog population. Not very likely. Coming back from a dog to a human, I think these events are going to be rare, but perhaps the best advice would be not to, for the meantime, not to pet other people's pets or dogs. And probably we're all keeping our pets indoors anyway. So when we take them for a walk, I think most people keep their dog on a leash and don't let them interact with other animals. If people do that, then highly unlikely the animal is going to either become infected or transmit the disease. And I think the other advice I would give is if someone unfortunately becomes infected and they have an animal, it would probably be better if they were to allow someone else to look after the pet while they're sick. That's all I can really say about that at the moment.
Thank you very much. That is all we have question-wise. But is there anything else that you would like to add or that you think would be salient for people to know?
Yeah, I think what the best thing to add is that I think everyone's feeling frustrated and hemmed in because we're all staying at home and the whole concept of keeping physical distance from other people is hard. I would say that we have to realize how important this is to stop the spread. If we are not in contact with other people, we maintain our distance and we limit the interactions we have directly with people, we will absolutely limit the spread. The thing I think people will find a little hard to understand is that I think many people are thinking, well, I'm already doing this. Why is the virus continuing to spread? And that's because this virus has a very long incubation period. It can be as long as 14 days. And so we only really started socially distancing from each other less than 2 weeks ago, really, in reality. So we actually have to wait longer than that 2-week period before we'll start to see any effect. So I would encourage people to keep it up and wear it as a badge of pride that you've managed to keep away from individuals. But I would also encourage people to talk, just like we're doing right now, Jonathan. Use things like Skype, Google Meeting Rooms, and Zoom to keep in touch with people. If you do that, you'll keep sane. You'll be able to talk to your friends and family and make this all much easier.
All right. But once again, thank you for your time. I really appreciate
fun chatting and stay safe and stay sane. Stay sane.
Absolutely, you too. All right.
Take care.
Bye,
Jonathan. Bye-bye.
That's it for part 1 of this 3-part video series. If you want to access the other parts, go ahead and find the links in the description down below. And with that, I will see you next time.